good to spend a little more time with you talking about some of these topics. And biochemistry is something I'm really passionate about, so I couldn't get away without at least talking about it some. And asking you to consider the fundamental basis of living systems, at least from a physical standpoint, has to do somewhat with biochemistry. And so I think it's a really important thing, and it opens up some really interesting doors. And I think, as Dr. Wood pointed out, I agree. I think we live in a world where we're starting to understand that biological change is something that is not only possible, but happens and is observable, and may be part of the way God designed the system. This concept of modularity seems to pop up in a lot of places, not just in genomics, but we see it playing itself out in terms of enzymes. And I'll try to point some of that out today. You know, the article from the 1970s by Dobzhansky, this article that was titled "Nothing in Biology Makes Sense Except in the Light of Evolution." I encourage you to read that article and at least get the perspective of what he was saying in the article, and what he meant by it. And the reason this article has come to mean something more to me over the last couple of years is I was asked to help Dr. Ralph Gilmore from Freed-Hardeman in his debate with an atheist by the name of Alex Rosenberg. And Rosenberg is a physicalist, meaning all that exists is physical. And one of his main drivers in his understanding of evolution in terms of the way he interprets the world is this phrase right here. This title was his mantra. He called it Dobzhansky's dictum. And he touts this throughout his books. And so I had to hear it a number of times. I can't remember, it was a few years ago in either Creation Research Society Quarterly or another one of the publications. There was an author that actually wrote hey, let's look back at that. It was 40 years since he wrote that article. Let's see if what he claimed to be true actually was true. And so it's an interesting read. I'll try to find that for you if you're interested. And this other quote that gets to me, and this, again, I just want you to understand, this is the mindset a lot of us are trained under, right? This is what a lot of my professors in graduate school believed. But then you have people like Francis Crick, and he says, biologists must constantly remind themselves that what they see was not designed, but rather evolved. And Dawkins and others readily admit that, look, things give the appearance of design. These things appear to be designed. But then many people go on to ask you to consider that what you think was designed may not actually have been designed. In fact, we're pretty confident that it wasn't designed, that it really wasn't necessarily an intended product. It just happened. And I have a problem with that line of thinking because it's asking me to go against what I feel like the weight of the evidence shows, and that is that this is a designed system. Even if we talk about the change, the types of change, and the amount of change, I think it's clear that even if you give room, let's say, for lots of change, what if that change was part of a designed system, a design mechanism? When we look at computer systems, we talked about this yesterday, just the idea that there are layers of organization in living systems, just as in computer systems there are layers. And these are two different layering models for the concepts of how computer systems are built. And I would like to suggest to you that this is not unlike a living system, that we're built in layers. And I showed you this the other day, just the idea that we have all of these different layers of complexity, one built upon the other. And yet we know, if we zoom in on kind of that cellular level, we know that life is in essence cellular, right? We consider that to be kind of the basic unit of life. You'll see that as some textbook definitions. So cells are really critical for a lot of reasons, and there's a lot going on here, right? We could walk through and discuss the importance of the membrane out here on the outside of the cell. We could talk about the mitochondria, which are the powerhouses of the cell. We could talk about this crazy thing that we call the endoplasmic reticulum and all the various things it does in terms of both protein synthesis as well as modification. And then we've also got things like lipid and carbohydrate synthesis work that may go on there some. You've got The Golgi apparatus, important in packaging things that are going to get secreted, doing some modifications, and then a number of other things. You've got lysosomes, peroxisomes, you've got ribosomes, which are massive RNA and protein complexes, which are some of the builders of the cell, right? These are the things that are making proteins. So we've got all of these different things in this little world that we call the cell. And the fundamental level is that we've got four basic kinds of biological molecules. Amino acids, which make up proteins, nucleic acids, which make up DNA and RNA, lipids, which are fats, fatty acids, steroids, all that, and then carbohydrates, which are sugars. And all of these molecules are in and of themselves discrete entities, but what we find out is, and I'll show you this here in a minute based on this chart, they all flow in together, right? They're all interconnected on some level, which to me is mind-blowing but yet cool at the same time. Biological molecules are interconnected. And so lipids and carbohydrates, for example, are synthesized and metabolized by proteins. All right, that's not a big deal. We understand that. Well, proteins are made by proteins and RNA. All right, got that? Well, RNA is made from DNA by proteins, and that's ignoring the fact that we also have to make the RNA nucleotides from amino acids and carbohydrates, right? So you got that process that also has to take place. Nucleotides, again, DNA and RNA made from carbohydrates, amino acids by a number of different protein enzymes. And so you've got this scenario here where our cells are using things to build other things that then are dependent upon other things to build more things, and it's a very big process that we're going to look at. Now this chart, as Dr. Wood said, this is kind of the wow factor, right? This is the whoa, there's a lot going on there. But it's also very understandable, and it's something that is really, really cool when you just step back and take a look at it. Now this map is freely available online. This is from Stanford University, Stanford's Pathways of Human Metabolism. And it's a really good resource. They continue to revise it, and so I encourage you to look it up and download it and learn from it. But what they've done here is they've tried to organize the various areas of metabolism, and this is humans, so understand you could draw a different map for other organisms, which are going to have some differences from us. But in this map, you've got over here, you've got kind of the lipids area over here, and then you've got the carbohydrate area here, and the amino acid area here, and the nucleic acid area over here. And they've got it drawn out like that, and then they've also got delineated that this is kind of the mitochondrial barrier here. So stuff in the middle of that is in the mitochondria, right? Especially things like the citric acid cycle, that big circle there. And then we've got down here the ATP synthase pump, things like that. All of that's taking place in the inner mitochondria within the inner mitochondrial membrane, or along the inner mitochondrial membrane, as some of these proteins down in this area are. What I want you to understand, though, is that these are not all isolated things, and we have feedback and we have interplay between the various pathways. It's hard to depict the level of it that takes place, but you can see it in a few places here as they try to show you the interconnections between both lipid metabolism and carbohydrate metabolism and amino acid metabolism, and yes, even carbohydrate and amino acid metabolism and nucleic acid metabolism. It's probably better not to draw this as a two-dimensional chart, but rather as a three-dimensional, complex, so to speak, because there's a lot of things going on here. But what I want you to understand is that this is fundamental to a living organism, right? If we're going to build an organism that lives on its own, we've got to think about all of the components it takes to get the job done, which is a lot. And how do all of those things interplay in such a way. This to me, talking about, why do we believe what we believe? This to me is fascinating evidence of a grand design, meaning all of this interplay between these molecules, so finely and delicately balanced. There are feedback mechanisms, there are control mechanisms. All of this interplay to me is not coincidental, it's not accidental. Right? This is a series of processes that have to take place. If this stuff doesn't work, we're in big trouble. right? There are a lot of diseases. There's a whole study of what we would call inborn errors of metabolism. And med students go crazy over this stuff because the med schools push the students to understand if you've got some obscure disease state, you've got to figure out what's going on. Here's a whole list of inborn errors of metabolism. Now, I was going to teach this to my pharmacy students at one point. I thought, I'll just look this up and see. And I found out, you know, there's like 20 some odd pages of lists of different things that can go wrong. And when those things go wrong, some of them, you know, people survive and they can have maybe a relatively normal life, but maybe they have some dietary deficiency. They need to have some supplements or something. But some of them can be very debilitating. Just simple errors in this pathway, which to me underlies the critical nature of how all of this works together. Take, for example, our discussion from the other day on this idea of the central dogma, DNA goes to RNA, goes to protein. There's what we would call causal circularity here. In other words, the products of the processes are needed to complete the processes themselves. And that's a fundamental concept in biological systems. A few years ago, was it Craig Venter? They took a small bacteria, they pulled the DNA out of it, they used massive DNA synthetic technologies to build a new chromosome, stitching it together, and then they put it in the bacteria, and the bacteria survived. And what'd they say? Right, the headlines read what? Hey, we made life, right? We made life. What they didn't tell you was they provided all of the machinery, right, to do all of that, and they swapped out the DNA, which is still amazing. I don't want to belittle it, because the fact that they were able to synthesize huge chunks of DNA and stitch all that together is an amazing task from a biochemical and genetic perspective. I think it's amazing. So I don't want to belittle it, but it's not quite creating life. So we just need to you know, realize we may have to temper that a little bit. For that organism to survive, it had to have all of those components in place, right? It had to have the ability to replicate DNA, had to have the ability to transcribe DNA, had to have the ability to translate proteins. All of those components were in the host cell that they inserted the DNA into. Is everybody following me here? Without that, right, if they just put that piece of DNA into a lipid bilayer, wouldn't have done anything, right? Just sat there. It was only because all of those systems were in place that that actually worked. But this idea that the products are required for the processes themselves is really fundamental and something we really need to understand on a very fundamental level. So DNA, I'm not going to go through this. We've talked about that. You understand the structure of DNA, the double helical nature of DNA, the idea of chromatin. So we've talked about all of those things. Just to review really quickly, the idea that the genetic information in the DNA codes for other levels of information. So ATG up here in the DNA strand becomes AUG in the messenger RNA, which becomes methionine in this protein sequence. Everybody understand that? Familiar with that concept? And these sets of three, get back to it. Those sets of three nucleotides we would call codons, and they code for different amino acids along the way. And so again, if you change a nucleotide, you can potentially change the codon that you get in your messenger RNA, which may or may not change the amino acid, right? Depends upon what nucleotide changed, where is it, what amino acid is it, etc. So some mutations, some SNPs, so to speak, are silent to the extent that we can tell on a protein level. Okay, so proteins, of course, you know, amino acids then are combined in long chains in a specific order as specified by the messenger RNA. Now, some of this is interesting because we're starting to talk about design change. We already know that there are systems that do this, right? So antibody diversity, how is it that your immune system has so many different versions of antibodies? Well, there's a mechanism built into our body for generating diversity in the region where these antibodies are generated genetically, so that in the B cells that are making the antibodies, they can make a bunch of different binding epitopes and identify a wide range of potential invaders, right? And there's a process your body goes through to make sure those antibodies don't respond to self, right? So you can avoid autoantibodies and autoimmune disorders. But just the idea that the information in the messenger RNA and the information in DNA specifies a protein is true, but we do have to hedge and accept the fact that there's some change that actually does occur, right, in terms of antibody diversity and things like that. Proteins can serve a lot of different roles: structural, transport roles, signaling roles, catalytic roles, many other things. They're multifunctional. Some of these proteins have more than one role. Sometimes we call that protein moonlighting, the idea that a protein may be this, but also do this. And so you'll see that very commonly. But another thing you'll see, and this is something that I talk with some of my colleagues with, is some people say, well, this protein does this. And you say, well, no, it doesn't do that. Let's say in the case of topoisomerases, I've been told by some, well, topoisomerases regulate neural development. Well, not exactly. Topoisomerases regulate topology, and topology changes may be needed for neural development. And so you could see how some people make that leap, but what is the topo doing? Well, the topo in the functional role is regulating topology. The consequence is it's going to allow or not allow, depending on whether or not it's functional, the use of that region of DNA. So most enzymes are proteins. A few are catalytic RNAs that we might call ribozymes, but generally speaking, when we say enzyme, we're talking about something that's a protein. These perform the work of cells. Why aren't more functions carried out by catalytic RNAs? Why do you think that is? This is a question for anybody. How many different nucleotides are there versus how many different amino acids are there? So amino acids, we've got about 20 different amino acids, give or take a few, right? You could have some modified versions, versus 4 nucleotides in RNA. So what would be the spectrum of catalytic properties of an RNA molecule with its 4 different nucleotides and different arrangements versus the catalytic capabilities of 20 different amino acids arranged in various arrangements? So you can see very quickly that having 20 different R groups, as our amino acids do, there's a lot more ability to have diversity of structure and function versus a catalytic RNA. Now, catalytic RNAs are pretty cool. In fact, I have to give credit to catalytic RNAs in my graduate work. When I was doing my graduate work on metal ion function in topoisomerase II, we were using a modified piece of DNA, and it turns out that type of modification is used a lot in RNA, catalytic RNA studies. And so I read a bunch of catalytic RNA studies trying to figure out how did they use this modification and what did they learn from it. And it was from those studies that I decided we could use the modified DNA to study something about how metal ions are used in the active site of Topo 2. So it was a completely different field, right? Completely different concept, but we were able to draw upon what they did to make a connection there. So I do give some credit to catalytic RNAs. They did something very cool for me in my graduate career. So the human genome encodes thousands of different enzymes, right? And who knows how many different versions of enzymes. Todd mentioned the other day how many different transcripts we get from the protein coding genes. It's a ridiculous number. And so what we know is that there's the potential for a lot of different versions as various exons and introns get spliced in and out. The ribosome is something I also like to talk about just briefly to note that this massive structure, this massive complex, is not just protein, but is RNA and protein. So to go back to our idea here, we've actually got to have both for this machine to work. And so 80 or so different proteins, maybe up to 100 different proteins, depending upon the complex, in 5 RNA chains. Some of these RNA chains are huge, right? They may be thousands of nucleotides long. Just to stop and think about the size of that in the cell and how many of these we need to do this job. This is a massive machine. But one of the other details that we gloss over sometimes when we teach the biochemistry of this is the whole idea of where do the amino acids and tRNAs come from, right? We have these tRNAs that deliver an amino acid, but we gloss over the fact that to get a tRNA that has a specific amino acid attached to it, you've got to have an amino-acyl-tRNA synthetase. In other words, we've got to have a whole other set of enzymes whose job it is to grab the appropriate tRNA and to pair it up with the appropriate amino acid. In other words, translation does not work if you randomly assort tRNAs with different amino acids. You've got to have a specific tRNA that grabs the right tRNA synthetase, that grabs the right tRNA and pairs it with the right amino acid. That's a really, really critical thing. In fact, there are diseases, by the way, talk about inborn errors in metabolism, there are diseases that result from mutations in amino-acyl-tRNA synthetases. So this is not an unheard of thing. What I want you to realize, though, is this was a big problem in the minds of some, at least, when they were trying to figure out how life could have originated naturalistically. Because this is a major issue, right? Going from DNA to RNA is simpler in concept than going from nucleic acid to amino acid. Because DNA and RNA, we've got this base pairing mechanism. It seems very elegant, but it also seems relatively simple in some sense. But to go from the DNA language to an amino acid language, that's wholly different altogether, because there's nothing inherent in the amino acid that specifies that it should pair up with that particular codon. That's why you've got to have the tRNA molecule itself. Hence the need for amino-acyl-tRNA synthetases to grab the tRNA, pair it up with the right amino acid so that it can go off and do its job. Does that make sense to everybody? It's a very, very basic concept, but it's also very important to realize this is another barrier to the idea of this thing having developed naturalistically. And it was recognized as such. In fact, Francis Crick realized that there was going to be a need for such a thing, and he offered what was called the adapter hypothesis. He probably wasn't the only one, I don't know, proposing something like this, but he proposed what's known as the adapter hypothesis for going from RNA to protein. James Watson wrote this in response to that concept. He said, "I did not like the idea at all. More to the point, the adapter mechanism seemed to me too complicated to have ever evolved at the origin of life." And now it may go to explain why some of these guys believed in the idea of panspermia, that life originated somewhere out there and was planted here, right? Because some of them recognize on a biochemical level, there's a real high level of sophistication to this system as a whole that doesn't just originate by accident, doesn't just originate by chance. And so, here's a structure of a particular tRNA molecule. And so that gets grabbed by an enzyme and paired up with a specific amino acid so that you can have the translation process work properly. So this is what helps us translate RNA to protein, right? Without this molecule, and not just one, right? You've got to have several different versions, because remember, we've got these codon loops at the bottom, 3 nucleotide sequences. So there's 64 possible combinations there. And you've got to have those to bring things together. All right. So how much does it take to do all of this? I think this is an interesting comparison just to give you a perspective. And so by the numbers, humans have about 3 billion base pairs. E. coli is about 5 million. Mycoplasma genitalium is about 580,000. Right, so one of the simplest genomes that I'm aware of, Dr. Wood, I don't know if that still holds true. That's probably about the smallest genome we know of. Mycoplasma genitalium is one of the smallest. Now, understand this is a parasite, so it has a host. It's living on a host, and it requires a lot from that host in order to survive. But just as a way of comparing what it takes to get jobs done. So in humans, we have over 160 genes that are associated with replication or repair, and over 30 involved just in replication or DNA synthesis alone. Mycoplasma genitalium still has about 30 different proteins involved in that. Transcription in us, about 40 different proteins. Mycoplasma genitalium, about 10. Translation, there's over 100 different proteins involved in that versus about 100 in Mycoplasma genitalium. Energy metabolism, about 70 different proteins and about 30 in Mycoplasma genitalium. So here we've got one of the simplest known living organisms. Again, it's got to live on somebody, right, to survive. It doesn't live on its own. So it's getting certain things provided for it, is what I'm saying. under those circumstances, it still requires quite a bit. In fact, based upon this particular study, they found that it requires about 382. So it has a total of about 400, maybe 480, something like that in terms of total gene count. 382 of those are required just to survive. Assuming it has all the nutrients it needs, you still have to have 382 genes just for that organism to survive. What am I saying here? What I'm trying to suggest to you is that on a fundamental level, there is a minimal component biochemically for something just to be alive, to survive, and to do various things that cells need to do, like grow and divide. And it's a pretty high bar, right? That's a lot of information. I mean, even if that cuts out, let's say that cuts out 200,000 base pairs, you're still talking about 300,000 to 400,000 base pairs needed for this genome, right? You're still talking about quite a bit of information that's needed on top of the fact that this thing can't survive on its own. So I just wanted to give you some thought about, how do we compare living organisms, and what would it look like if we pared this thing down to as simple as possible. And maybe you can make one simpler than this. Maybe there are things you could add or omit from this particular organism. But again, you have to remind yourself that this thing isn't free living. Now, there are bacteria that can live freely in the environment, right? So E. coli is at about 5 million. I don't know. There may be some that live on just a few million. But the point is it requires more in terms of information and capabilities in terms of the enzymes and metabolic pathways for an organism to kind of live free and survive on its own. So I just think that's a really interesting thing to step back and kind of consider, what does it take? So again, cellular function is very dependent upon complex pathways. There's a lot of interplay back and forth. They're highly regulated, so there's feedback mechanisms, control mechanisms. And then we've also got signaling pathways inside and outside the cell to communicate, control, coordinate, etc. You think about feedback and control. When we go here in just a little bit, we'll go eat lunch, right? Then there's going to be a whole process going on as our body starts to digest that food, free up the carbohydrates, the lipids, the proteins, the amino acids from those, absorb those from the digestive tract, distribute them through the bloodstream. The body starts signaling, right? Our glucagon levels are going to drop, our insulin levels are going to go up, and cells are going to respond to that. They're going to dynamically respond to the environment to say, oh, it's time to take up glucose and start metabolizing it. Signals will go into the nucleus. You'll have changes in gene expression. You'll have production of enzymes and proteins. You'll have energy metabolism genes going on. You'll have energy storage taking place, energy production taking place. All of these things going on. It's all orchestrated, and you don't have to even think about it, right? You don't have to tell yourself, digest, right? Metabolize, right? Absorb, excrete, right? So we just, all this stuff takes place. And that to me is fascinating. We think about some of the pathways, and I'm going to point here to this for just a minute. If we bring food in as glucose here at the top of this pathway, what does it do in our body, right? It's going to circulate through the bloodstream. It's going to cause a rise in our blood sugar levels, right, which is going to cause insulin expression. And if you happen to have appropriate biological response to insulin and you're not diabetic, the signals are going to go to your cell to tell your cell, we need to take this glucose up. Glucose is going to go through a series of events and pathways to be metabolized. In fact, this is the pathway. So this is a simplified version, okay? So understand, we've omitted some of it just for sake of time and clarity. But the general idea is that insulin is going to bind to this little guy here that we're going to call the insulin receptor. So this is what we call cell signaling. So when you think about signaling in a cellular context, there are some things in neurons that are electrochemical in nature, but a lot of signaling in our body is more about actual physical molecules circulating around, binding to receptors, and then those receptors telling the cell, hey, we're getting this signal, we need to respond. And so the insulin receptor gets phosphorylated, turns on what we call the IRS, not the Internal Revenue Service, but the insulin receptor substrate, very creative name. And that's going to activate here. We've got PI3 kinase, and we're going to make PIP3. PDK1 is going to be brought together. Oops, excuse me. Back up. So PDK1, AKT, et cetera, et cetera. So you're going to have pathways here that go on within the cell. You have growth and survival effects. You're going to have transport of the glucose transporter to the cell surface so that you can bring glucose into the cell. All of these things are going to be taking place, right? Just because insulin bound to a receptor on the cell surface. And that's one pathway, one example of one pathway out of many, many, many different types of pathways that are going on all the time. Right? We've got several different classes of receptors. In fact, receptors are a huge area of drug targets, right? Because we can manipulate receptor responses in a lot of different ways. A lot of the drugs that are used in our clinics and hospitals target various types of receptors and turn them on, turn them off, you know, and modulate function somehow. So there's a lot going on there, and to me, it's really, really fascinating. It's really, really cool because, you know, the journey just keeps going, right? Glucose gets phosphorylated in the cell to glucose 6-phosphate. Glucose 6-phosphate gets converted to fructose 6-phosphate, which gets phosphorylated again, fructose 1,6-bisphosphate. That gets cleaved in half to make dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. This DAP here gets converted to GAP. The GAP's going to go down here, 1,3-bis-glycerophosphate, 3-phosphoglycerate, 2-phosphoglycerate, phosphanol pyruvate, and ultimately pyruvate's going to get in here to the mitochondria. It's going to be taken in and feed carbons into the citric acid cycle. All of this process is harnessing electrons, which are going to go to the electron transport chain. The electron transport chain is going to generate a proton gradient. The proton gradient, as it's dissipated, is going to turn an ATP synthase motor, which is going to make energy so that you can stay awake, right? All of that stuff, it's just happening. But then there's the interplay, right? Because certain things here are going to feed in and out of these various pathways. We've got places where fatty acid metabolism is going to feed into the citric acid cycle. It's going to feed into carbohydrate metabolism. We've got places where amino acid metabolism can feed into this pathway. Guess what happens if you don't take any sugar in? Your body starts using other things to make what it needs to make the energy. You'll burn fat. You'll burn proteins, right? You starve long enough, and you'll burn proteins up. It's all designed to interplay. What happens when we need to make DNA so that we can have nucleotides for DNA and RNA synthesis? Well, we're going to draw upon a number of different amino acids. We're going to draw upon certain metabolites of this pentose phosphate pathway here. And so we're going to take our ribose 5-phosphate over here. We're going to take certain amino acids over here. And we're going to feed them into this pathway. And what happens when we break the DNA down? Well, we metabolize the nucleotides into their components, and then that can all get recycled too. It's really an amazing process. If you think about the interplay and how, while we think of this as here's lipids, here's carbs, here's amino acids, there's nucleotides, they are all separate entities, but they can be interchanged, which is really, really cool. And this, to my knowledge, this is true throughout all life forms, right? That we have that interchange. Now, some organisms can do more of that than others. For example, there are certain fats that we can't make, right? So we have in our diets a requirement for omega-3 and omega-6 fatty acids because there are certain fatty acids that derive from those, eicosanoids in particular, that we cannot synthesize on their own. So we got to have some of that in so that we can package it in the right phospholipid context so that later we can utilize it in various pathways. So there's a whole lot going on here that I want us to have an appreciation for the elegance of the way this system works. And of course, you know, what happens when it goes wrong, right? When this system doesn't work, there are a lot of problems that come from it. And so it to me reminds me of the fact that God designed this system. It's very robust, but it's also really critical. And when it doesn't work, that's a bad thing. Okay. So we talked about topos the other day for a minute. I wanted to bring up, how much time do we got left? Okay, good. We're doing great. So we walked through all this. I wanted to show you this for just a minute to get you to consider something about enzymes and proteins in living organisms. Now, my background is not comparative genetics and things like that. I'm trying to learn those things, and I'm going to sit at Dr. Wood's feet and try to learn as much as I can about how to do those types of studies, because I think it's really critical to understand some of that. But just my understanding from my own field, in looking at these enzymes, is very interesting. In the topoisomerase realm, we generally think of there being about 5 different families, and one of these is actually quite new. When I was being trained, there was really 4. So 1A, 1B, 1C, 2A, and 2B. So you kind of see the division here. So we got the type 1 enzymes and the type 2 enzymes, with the broad distinction being type 1 enzymes cut 1 strand, type 2 enzymes cut 2 strands. Now, it's interesting that the 1s and 2s ended up the way they did, and really what happened, if you go back to the initial discoveries by Jim Wong, what he found in the 70s was a protein he called omega protein, which ended up being topoisomerase 1. And thankfully, he called it topo 1, and it cut 1 strand. And later, they found, guess what? A topo that cut 2 strands, so they called it topo 2. And that wasn't necessarily an intentional, like we're planning this out. It was more just happenstance, and they kept that kind of approach and nomenclature since then. So what you'll find is that there's topo 2s, there's topo 4, there's topo 6, there's topo 1, there's topo 3, and now there's even a topo 5. So the odd numbers are always type 1 topos, the even numbers are always type 2 topos. So it works out. It's kind of nice. So, but again, I don't know that anybody really intended that during the process. As humans, we actually have 6 different topoisomerases. We have topoisomerase 2 alpha and beta. We have topoisomerase 3 alpha and beta. And we have a topoisomerase 1b and a topoisomerase 1b mitochondrial, which is a truncated form of the topoisomerase 1b. So we have 6 of them. Some organisms have 2. Some organisms have 4. Have various different numbers of them. What's interesting to me is something about the various distinctions here. Notice the second column, or third column, I guess, linkage. So when the enzyme cuts the DNA, one of the things we didn't talk about the other day is that it actually grabs onto the DNA covalently. So it doesn't let it go. And this is part of preventing a double-strand break in the DNA. So it grabs the DNA and it holds on to it. Now, if you're a type 1 topo, you only have to grab 1 strand, right? Because you're only breaking 1 strand. If you're type 2 topo, you actually need to grab strands on either side so that the DNA cannot just freely float away. And what we know is that that connection is made, for example, in type 1As on the 5' end of wherever the break is. In 1B and 1C, it's at the 3' end, and in the type 2 enzymes, it's on the 5' end again. So we have different types of connections. What's interesting is all of these use a tyrosine as the active site amino acid. So it's the hydroxyl group of a tyrosine amino acid that's actually grabbing onto the DNA in all of these, every case. But then there's different mechanisms. So notice type 1A, it's what we call an enzyme-bridged strand passage versus the 1B and 1C, which are controlled rotation. So this is really hard to wrap your mind around, but if you think about the rotation one's easy, right? Because if you got 2 strands and you break 1, you ought to be able to just twist, right? So controlled rotation, that makes sense. The enzyme-bridged strand passage is a little weirder. It's kind of like it goes like this, if that makes sense. It's like it breaks one and then it passes around. So it's a little bit harder to visualize what's going on there. Compare that with our double-strand passage mechanism. The type 2 enzyme does a double-strand passage mechanism, and it either leaves a 2 base pair or 4 base pair overhang, depending upon which type. Notice also there's difference in the requirement for cofactors. Some of them require ATP, magnesium, just magnesium, or none. Gyrase is really interesting in this. It can actually actively positively supercoil DNA, right? So, wait, I said that backwards, didn't I? Yeah, so reverse gyrase, positively supercoiled DNA. Did I get that right? Make sure I got that right. So then gyrase down here actively negatively supercoiling DNA. So it's really an interesting thing versus some of these other enzymes that are really, their activity appears to be more focused on relaxation of the strain rather than making actually inducing torsional strain, they're actually alleviating it. But what we find is that some of these organisms that live at really extreme temperatures, for example, or under some kind of extreme condition, may actually have a need for inducing supercoiling to keep the DNA from basically denaturing. Does that make sense? So there's really a lot of interesting things going on, and I think there's a lot of cool stuff there. But you can see there, I gave some representative species where some of these are found. What's interesting is that Topo 5, to my knowledge, this is still the only known Topo 5. It's found in one organism. And it's different from type 1b and type 1a enzymes structurally. So it's its own group. You know, it's kind of like, hey, look at me, I'm really cool, I'm on my own. You know, you compare that with things like the type 2b, topoisomerase 6. Or even within the type 2a group, gyrase and topoisomerase 4 in bacteria are quite a bit different than the structures we see in eukaryotic versions. So the bacterial versions tend to be tetramers, heterotetramers, A2B2 tetramers, meaning they got 4 subunits, 2 copies of 2 different subunits. In us, our topoisomerases are homodimers. Same protein, 2 copies. And so it's really interesting to kind of wrap your mind around that. And depending upon who you read, there are a minimum of probably 6 to 8 independent origins, and maybe many more than that, of topoisomerases based upon structural considerations and phylogenetic considerations. What am I saying? I'm saying if evolution is true, you have to have some way of accounting for independent evolution of multiple types of topoisomerases all independently. All converging, by the way, on the same fundamental mechanism, which is really, really similar across all these organisms. But the structures of the enzymes are so different that most evolutionists would probably say they could not have evolved from one another, but instead converged, evolved in a convergent manner on the same kind of solution. So what I'm suggesting to you is that what I see in the pattern of topoisomerases is that God used independent designs in various organisms to accomplish what needed to be accomplished. And we find that each of these enzymes is well suited to its environment and to the particular organisms that it operates in. So to me, I think it's a really cool thing to consider as we look at that. Now, Topo 5 would be what we consider to be an orphan gene. In other words, it's present. Orphan comes from the concept of orf, or open reading frame. And an orphan is a gene that you find in a very limited subset of organisms, right? Maybe one or a very few number of organisms. And it's not found, let's say, anywhere else. And so what can we learn from orphans? And this is an area that I would like to learn more about. And frankly, I don't know much about it. But my understanding is that the more we sequence genomes, the more and more we find there are unique orfs, or open reading frames, in those organisms that are not found in other organisms. What does that mean? Well, it means that there are some proteins that we find that are predictably similar to what we find in other organisms. But then there are some proteins we're finding that are really unique, and we don't find other places. And that number continues to grow. And so the total number of proteins that we find that are similar is kind of static, right? Because there's a group of those, and we know we expect to find those. But we have this growing number of proteins every time you sequence a new genome that you find that are unique. And so these orphans continue to grow. And they are very interesting to me. And it may be that we find, you know, as you sequence more, some of those orphans may actually be found in other organisms here and there. But the point still is there's a lot of them. And the question is, what can we use that to learn? And there's a lady at the University of Missouri named Tenn, who's doing some work that I think is really interesting. And if you haven't looked up her work, I encourage you to. I put 2 references up here, and I'm not going to get into discussing it at this point. But I think she's got some interesting ideas about how can we use these taxonomically restricted genes, or orphans, to understand the relatedness of various organisms. In other words, could this be used to complement what Dr. Wood and others are doing on maybe a biological level or a fossil level with various bone structures? Can we use maybe some of these genes to actually look into, are there ways to identify groupings of organisms based upon the essential genes? So she's put some of these things together. I think it's interesting. I don't know enough about it to really critically evaluate it very intensely, but I think it's worth taking a look at and considering. So I encourage you to look into that. There's a number of things out there on orphan genes and what they can imply for this discussion. So cells, certainly the basic unit of life. Biochemicals make that up. Biochemical systems are highly complex and interdependent, as we talked about. But then the open questions to me are things like what Dr. Wood ended on, and that is, how have these systems changed since the creation? And how much of that change is through what we might consider natural processes of mutation and selection versus maybe more designed processes within the system itself? And I think that's a really fair question to ask. A lot of not only the genome but also proteins appear to be very modular. And so that modularity may actually lend itself to certain types of changes. So again, if we use computer and engineering analogies, can we learn more about the system? So assume the system is designed up front and use that to frame your questions. Do you ask different types of questions than if you assume the system evolved? And I think you do. I think you'll ask a different type of question when you approach it like that. And frankly, I think we can ask better, more probing questions, right? When we talked about the other day the concept of junk DNA, some people still defend it, some people do not. But I think we can look at these non-coding regions and ask, is there function? And it appears, the answer appears to be coming back again and again, yes, it does something. But what it's required us to do is change how we think about what function is. In other words, function doesn't mean it makes a protein. Function may mean it serves as a binding site, it's a scaffolding point, it's a organizing point, it's a, et cetera. And I think the question to me is still open, and I love to hear more discussion on retrotransposons of how do some of these mobile genetic elements play into this to allow us to adapt to various circumstances and situations. So anyway, there's a lot there. I'm going to stop so we can have a few minutes to discuss before we head to lunch, but thank you all for your attention today and putting up with me again. So thank you.